नमस्कार दोस्तों, स्वागत है आपका भारद्वाज गुरुकुल में, जहां सीखना होता है आसान और मजेदार। आज हम क्लास 10 ICSE बायोलॉजी के एक बेहद जरूरी चैप्टर को आसान भाषा में समझेंगे, स्ट्रक्चर ऑफ क्रोमोसोम। तो चलिए शुरू करते हैं वो सफर। In this video, we learn what are chromosomes, mitosis and meiosis, nucleotides, DNA and RNA, genes, cell division, about the stages involved in the cell division and the process of new recombinant or variations. How do cells divide? How are daughter cells formed? Why do we look like our parents? Why are we different from our parents in some aspects? So let's begin and answer all of these questions in this video. Chapter Structure of Chromosomes, Cell Cycle and Cell Division. Contribution of some scientists. Walter Fleming discovered chromosome in animals. Rosalind Franklin discovered the shape of a DNA molecule. Watson and Crick proposed the double-stranded helical structure of the DNA molecule. Define the following. Karyokinesis. Process of division of nucleus is called karyokinesis. It is of two types, mitosis and meiosis. Cytokinesis. Process of division of cytoplasm followed by karyokinesis is called cytokinesis. It is specific in animals and plants. In animals, it takes place by the formation of depressions or constrictions in the cell membrane somewhat in the middle of the cell. And this constriction further deepens to divide the cell into two equal and identical halves, which are referred to as daughter cells. In plants, the cytokinesis is completed by deposition of cell plates somewhat in the cell. The cell plate further extends towards the boundary and divides the plant cell into two equal and identical halves. Meiosis. The process of division of nucleus twice is called meiosis. As a result, 4 daughter cells are formed, which are haploid and are found in the reproductive cells. It is completed in 2 steps. First division is the reduction division, and the parent cell is divided into 2 halves. The second step is equational division, and it makes each cell division split into 2 with the same number of chromosomes compared to the parent cell. As a result, by the end of the division, 4 haploid cells are formed. Mitosis. It is the process of division when nucleus divides once to produce only 2 daughter cells which have the same or identical number of chromosomes as compared to the parent cell. It is found to occur in somatic cells, that is the body cells, and in the reproductive cells also. Chromatin. 2 constituents of chromatin are DNA about 40% and histone about 60%. What is a nucleotide? It is a unit of DNA composed of phosphate, pentose sugar and a nitrogenous base, which are complementary to the nitrogenous base of the other strand of DNA attached by a hydrogen bond. Example, A double bond T and C triple bond G. Write the full form of RNA. Ribose nucleic acid, and DNA is deoxyribose nucleic acid. Its components are phosphate, sugar and nitrogenous base. Purine, the nitrogenous bases with 2 nitrogen carbon rings. Pyrimidine, the nitrogenous bases with 1 nitrogen carbon ring. Histone, the protein that helps in coiling and packaging of the DNA molecules into structural units called nucleosomes, is called a histone protein. How many histone proteins are wound by 2 rings of DNA? 8 histone proteins are wound by 2 rings of DNA. Such a complex structure is called a nucleosome. In order to fit approximately 2 meters of DNA within the nucleus, it is condensed in the form of coiled and supercoiled chromatin. Chromatin cannot undergo the process of duplication as it is highly condensed. Hence, it is to undergo the process of cell division to form the coiled condensed chromosome to undergo the process of replication. Functions of centromere. Centromere attaches the sister chromatids. Spindle fibers get attached to the centromere of chromosomes or chromatids at centromere. Helps in separation of sister chromatids during anaphase that helps to form 2 separate chromosomes. Genes. Nucleotides are present in DNA, and DNA is present in genes which help in transfer of characteristics from parents to offspring. In males, we have 22 plus X and 22 plus Y chromosomes, and in females, we have 22 plus X and 22 plus X. So, together in males, we have 44 plus XY chromosomes, and in females, we have 44 plus XX chromosomes. The cell undergoes the process of cell division with the same number of chromosomes as compared to the parent cell, or 4 cells with half the number of chromosomes compared to the parent cell before undergoing the process of cell cycle. The cell passes through the preparatory phase known as the interphase. Interphase is also called as the resting phase because the changes the cell undergoes are not visible by simple magnifying devices like the light microscope. Though it is a highly active phase of the cell division, it is completed in the following 3 phases. G1 phase, synthesis phase, growth 2 phase, and mitosis phase. Interphase, growth 1 phase, protein and RNA get synthesized. Mitochondria and chloroplast divide. Cytoplasm doubles in volume. The synthesis phase, synthesis of DNA continues. Growth 2 phase, proteins and RNA synthesis continues and enters into the mitosis phase. What is the resting phase of the cell? Why is it the most active phase? Draw a diagram of interphase of cell cycle. Which phase comes after interphase? What is the characteristic of that phase? So these are some important questions whose answer you should be knowing. New recombinants is the structure formed due to the interchange of chromosomal segment between two non-sister chromatids of homologous chromosomes in meiosis. The crossing over of two non-sister chromatids forms new recombinants. Two homologous chromosomes are chromosomes from two different parents with same shape and size. M phase comes after interphase. M phase, also known as mitosis phase, in which nucleus divides only once with same number of chromosomes as compared to the parent cell. It occurs in somatic and reproductive cells. So you can see the diagram of the interphase state of the cell where centrosome with centriole is visible, cytoplasm is visible, chromatin fibers is visible, and the nucleus and the nucleolus are also visible. Now let's move on to the next stage, that is the prophase. Here you can see the nucleus, the chromatin fibers are arranged, the nucleoplasm, centrosome with centriole, nucleolus, and the centromere becomes visible during the prophase. Now in the second step, the centrosome with centriole starts moving towards the opposite poles, as you can see in this animal cells diagram. Next, chromatin becomes short, coiled and condensed, forming the chromosomes. You can see in the nucleus the chromosomes are formed which were earlier chromatin. In the third stage, chromosome divides vertically into two chromatids. Now the chromosome splits vertically and forms these chromatids. Nucleolus disappears and nuclear membrane starts disappearing. Spindle fibers appear from the two opposite poles. You can see in the diagram the nuclear membrane is slowly disappearing and the nucleolus has already disappeared. Next, the spindle fibers get attached to the centromere of the chromosome. The spindle fibers have arisen and the centromere of the chromatids get arranged over there. The chromosome gets arranged on the equator of the spindle. The arms are facing their respective poles. So the arms of the chromatids are facing the respective asters. Now the third one is the anaphase, which is also referred to as the shortest phase. Centromere splits but chromosome stays at equator of the spindle, as you can see in the diagram. Chromosome starts moving towards their respective poles. Centromere acts as the arrowhead towards their respective poles and arms facing the opposite pole. So they start moving towards their respective poles. A furrow starts appearing somewhat in the middle of the cell. Cell constricts. So this furrow has started to develop as this is the animal cell. As you can see, the furrow is becoming deeper and deeper. The cell constricts in the middle of the cell. Spindle fibers disappear, nuclear membrane is formed, chromatids turn again into chromatin fibers, and the two daughter cells are formed and are secreted by a protoplasmic septum. The furrow deepens and separates the parent cell into two daughter cells. As you can see, they are identical and have the same number of chromosomes. And if it would have been a plant cell, then the cell plate starts moving towards the periphery and the division takes place. Synapsis means pairing. Define the process of formation of new recombinants by using the following words. So these are the keywords which are given: pairing, non-sister chromatids, crossing over, chiasma, interchange of chromosomal segment, and homologous chromosomes. So now let's look at the definition. During pairing of two homologous chromosomes, two non-sister chromatids join at a point called chiasma where interchange of chromosomal segment takes place. This leads to crossing over and forming new variations or recombinants. Meiosis helps in the formation of variations or recombinants. This process helps in the adaptation to the ever-changing environment and development of the race. Quick tips for success. The concepts of chromosomes are not just theory, they describe the world of cells. Mastering them makes you understand how life starts. उम्मीद है आपको ये वीडियो पसंद आया होगा और स्ट्रक्चर ऑफ क्रोमोसोम अब पहले से काफी आसान लग रहा होगा। अगर हाँ तो वीडियो को लाइक शेयर जरूर करें और भारद्वाज गुरुकुल को सब्सक्राइब करना ना भूलें ताकि आप कोई भी जरूरी चैप्टर मिस ना करें। नीचे कमेंट में बताइए कौन सा चैप्टर आपको अगला चाहिए। मिलते हैं अगले वीडियो में, तब तक के लिए पढ़ते रहिए और आगे बढ़ते रहिए।